I will have very easy job now because Flo and Megan explained most of the background, but I still going to have some of their slides here. So this just shows the delta 15 annual distribution around Maui, and as you can see that most of the largest amounts are around injection wells, and the amount of delta 15 nitrogen dissipates as you go further away from the source. So this is a strong indication that nutrient-rich water comes in from the wastewater treatment plants. And since our last meeting, I published a paper in the Brahman Seal, and in the table you can see that the highest delta 15 values that anybody recorded in the world is published, and it goes up to 50, it's mostly things long. Again, you can see the plume here, and it seems like there's a highway rushing down from the line to the treatment facility all the way to the Bay, and the plume goes out. So what are the effects of high nitrogen in the water? It increases the primary productivity of the coastal systems, and it can lead to opportunistic microalgal blooms, which in turn diminishes water column oxygen levels, negatively affects the benthic and fish communities, increases microbial abundance. And this increased microbial abundance can lead to skin infections, and anecdotal evidence that the frequency of these skin infections increasing kidney, there's bubbling water, blackness causing alcohol, indicating toxic conditions. So the questions we ask is, are there increased microbial levels? Because Megan's study really showed that there was lots of nitrogen. And what type of pathogenic microbes are present, and what are the sources? And we are actually looking at all three pathogens. We are looking at viruses, we are looking at protozoans, and we are looking at bacteria. And we are using the organized chain reaction to pin down the exact molecules. So that there are a couple of indicated microbes that Jody kind of steered me towards. And the first one is the general Bacteroides, which will tell us what's the overall microbial load. It's a non-specific indicator. It will pick up bacteria from domestic animals as well as humans. And once we determine that, we can look at specific human microbial loads. And there's human-specific Bacteroides, PCR assays, and also there's human-specific archaea of bacteria PCR assays, and there's also human-specific viral PCR assays. So the sources of microbes can be many. It could be a wastewater treatment plant, like a HCRI, it could be storm runoff, it could be floating septic systems, and a lot of private septic systems on Maui, on the Leeward side. It could be wildlife, pets, agriculture. So which one is the source? And that's the central question of our investigation. So to get a first-hand experience, we took a field trip to Maui, and we went to the wastewater treatment plant, and I sampled the influent and effluent. I went to the freshwater sieves. I was trying to see the sieves, but it was too murky, I couldn't see them. Hopefully next time I'll be able to see them. And we came back to Oahu and went back to the lab and ran some PCR for the presence or absence of pathogenic microbes. And so I extracted DNA from the PCR. And so while we were at the wastewater treatment plant, one of the workers told us they were chlorinating the effluent. And on our first test, we looked at the Methanobrevibacter CPI, which is a human-specific, and we couldn't detect any. We couldn't detect the presence of this bacteria in the effluent. And we had We saw the same thing with the specific vector release. There was no detectable human-specific vector release in that one. However, when we looked at general vector release, which can come from domestic animals as well as humans, we saw a big signal. So, in conclusion, it seems like that the chlorination of hyena is somewhat effective, but does not eliminate all bacteria from the effluent. And after hearing that the chlorination is not persistent, then it's not really a surprise. So the other question we have to investigate is how good these markers are, what are the sensitivity and what is the specificity of these markers. And so sensitivity is the ability to detect the target when present, the specificity is the ability to rule out the target when absent. And we also have to look at the limit of detection because we are going to collect seawater from the bay and the bacteria are going to be much more dilute than the ones we picked up from the effluent. So we have to do some experiments to find out how many fold dilutions are going to work. To address the specificity, I went on a group hunt trained by Jody. So I collected and tested so far 13 sheep, 5 horses, 3 dogs, 1 donkey, and 1 cat. And so I'm going to show you some of the preliminary results here. So when I looked at the Mechanobrevibacter, which is a gluten-specific indicator, I had 2 false positives in sheep number 1 and sheep number 6, and this is a positive control. It's just this wire. I got a similar picture with the human polyomavirus. At this time, I got positive in sheep number 4, sheep number 10, sheep number 11, and this is a positive control there. I'm pretty sure it's not a contamination problem because these sheep are different from the previous one. And of course, when I looked at the general vector release, I expected to see that each sample would be positive, and that's what I got. So the general vector release would pick up non-human fecal bacteria, and luckily it shows that it's very sensitive. I can pick up fecal bacteria from all the samples. And when I looked at the human vector release, the human-specific vector release, I got only 1 positive here in the sheep number 4. Although the size looks a little bit different from the positive control, so I will have to go back and confirm that this is really the same vector release or maybe just some non-specific PCR. But, so in conclusion, it seems like some microbial source tracking markers work better than others, and the importance of our work is that we really tested these markers in the wild. Some of these markers, or some of these bacteria that live in the GI tract of animals actually can live on the surface of soil because they have a toxic environment. So the specificity of these markers would be different in the wild from the meat. And so we have to consider the potential of contaminating sources. Are there any sheep or goats or cattle or horses from the area that we are going to study? Also, we have to do some DNA sequencing to confirm, and hopefully the next step when we start doing the quantitative PCR and we're going to use TACAM probes, it will eliminate false positives because the probes will hybridize to a certain sequence, and we are going to be able to find out which are the good indicator bacteria. And it seems like from my preliminary studies that perhaps the human-specific bacteroides, like bacteroides, theta-iodine, or some other specific bacteria will be a good indicator. So our plans for the next 2 months is to carry out the first pool sampling in February, continue collection of group samples for specific studies, start testing with PCR assays, with TechNet probes, and carry out lead-up detection experiments with the first order samples.